Okay, so now I want to move into talking about what are the steps involved in genetic epidemiology research at a very high level, of course. So the first step is to establish whether or not there is some genetic component related to a disease or disorder that we're studying. And I'll discuss how we do that in a moment. The next step is to establish the relative size of that genetic effect or relationship in relation to other sources of variation with respect to the disease risk. And then the third step is to identify the gene or genes responsible for the genetic component if we're able to do that. So how we do all of this is achieved through a myriad of ways, consisting of family studies, which I'll discuss shortly, and population level studies. So key questions that we ask when we're looking for genetic etiology of a disease are, first of all, does the trait aggregate or group in families? And the next is it inherited from parent to the offspring? What is the best mode of that level of inheritance if it is inherited? And then which chromosome carries the gene that's associated with that inherited trait? And then once we know the chromosome, we then can try to determine the gene on that chromosome and then the specific variant of that gene or genes that is associated with that phenotype, and then what genetic product is altered specifically that leads to that phenotypic presentation. So as you can see, we go from high-level relationships of family patterns all the way down to the molecular level, if possible, to understand if there is a genetic relationship, what is the specific genetic process involved and the product from it. So I'm illustrating here a figure that goes back to a paper from 1999 to illustrate the relationship between genetic epidemiology and molecular epidemiology and then traditional epidemiology. And I didn't technically define molecular epidemiology, but it is essentially a broader field than the genetic form of epidemiology in that we're looking at the molecular level factors that are related to disease and disease patterns. So what this figure is illustrating is the relationship among these three different fields, which again are highly related to each other. And the approach is used to determine genetic patterns. And we'll walk through family aggregation in a minute. But basically, if there's a disease that we're studying and it doesn't appear to be any family aggregation, then we move straight into the traditional form of epidemiology where we're now going to look for other factors that are potentially associated with that disease, other risk factors that aren't genetic in etiology, right? So maybe looking at nutritional factors or environmental factors. If there is appearance of a family aggregation, then we move on to doing Mendelian segregation studies. And if those fail to suggest a genetic pattern, then we can move into looking for other molecular markers that could be related to that disease. And if we find them, then we are going to study the genotype and the environmental interaction. If we do find a Mendelian segregation relationship, then we go into what's called genetic linkage, which I will introduce shortly. And if genetic linkage is demonstrated, then again, our ultimate goal would be to identify the responsible DNA for that particular problem or phenotype. And what's interesting about this figure, again, is it's from a paper dating to 1999, so it's well before the human chromosome was mapped. But people were thinking then, and much earlier than 1999 as well, about genetic patterns of disease. So then this is a figure from the post-genome mapping, and this takes those ideas I just showed and puts it into a much cleaner process because we're now able to identify locations on chromosomes. We can do other things that we could not do before. And I'm not going to walk through this in detail because I haven't introduced these concepts yet. So I recommend you refer back to this after I introduce them. But these are the kinds of studies we would perform, or analyses we would perform, to answer these specific questions. So if, for instance, we're looking at what is the best mode of inheritance of a specific disease, we would perform what's called segregation analysis. If we want to know where on the gene is the specific variant located, we do linkage analysis. And if we're interested in how does the gene control risk, or if there is a gene-environmental interaction, we will do a combination of those sort of studies. So this is the general flow of genetic epidemiology in terms of the research flow. So with any research, we always begin with descriptive analyses or descriptive approaches. And essentially, we've discussed those methods already in the last 7 weeks of this course. So we would be looking at disease characteristics at population level, and looking perhaps at person-level characteristics as well as population-level characteristics to understand this disease patterns. The next aspects of the flow of research are what we're going to talk about now. And I'll walk through all of these shortly, but I want to point out that these first 4 aspects of this flow of research do not require genetic or genotype level data. So these level of studies had been done well before the mapping of the human chromosome. It isn't until after the mapping that we were able to do linkage and GWAS studies, which I will discuss. And then the final point I want to make about this is the flow of epidemiologic research doesn't always occur in this pattern. Sometimes aspects of this flow, if you will, aren't necessary, in that a researcher or data already exists for the researcher that precludes the need for doing some of these studies. Okay, so I want to first talk about familial studies or familial clustering studies. I want to talk about family aggregation studies, or family aggregation as a concept when exploring genetic relationships with diseases. So we think of family history as an independent risk factor when we're performing family aggregation research. We look for we look at family history, and we look to understand if there are commonalities among members of a family in terms of also the presentation of a disease, to understand if there's aggregation within the family. So we collect a myriad of things, certain elements. We, for example, will look at the age of onset of the disease that we're studying among family members to understand if there's a genetic pattern going on. So there are a couple of examples of this in our textbook. One had to do with retinoblastomas, the unilateral versus the bilateral forms. And it's now well understood that the bilateral form of this retinoblastoma, bilateral meaning it's occurring in both eyes, is associated with, has a genetic component because of the way it presents, in that all members in the family will present with that phenotype early on in their life. In contrast, the unilateral form of retinoblastoma is such that the presentation is later on in life, and suggests therefore that there's not a familial relationship. Another way we look at relationships in families is the degree of the relatedness of affected individuals in the family who have the disease. So we'll look at people who are first, second, third generation members and look at whether or not they have the disease. So that's a very simplistic approach, but again, this is the initial way of looking for patterns of familial representation of a disease. And then obviously we'll look at the number of affected individuals in a family. Now, there are multiple limitations with family aggregation approaches. The main one being that not all diseases that are genetic in nature or have a genetic etiology develop rapidly. So you can look at the presentation at in a family level, but not all people may present with it because they may not have been alive long enough. And obviously these levels of research don't tell us which specific genes are involved. Again, that's something we need to do, or we need to do other studies, which I'll discuss later, to be able to understand. And these types of studies don't separate environmental from genetic causes of diseases. Now, when we do familial aggregation studies, we calculate what's called the relative risk ratio, which is the measure of the strength of the familial aggregation. And this should be the lambda sign. And I know on our slides it shows us that, but when presenting it, it doesn't. I'm not sure why. But nevertheless, the relative risk ratio is the measure of the strength of the familial aggregation. Now, the prevalence of the disease in the relatives of a family grouping is important, right? So the more prevalent the disease is in the family, the more people you have to study. And that'll affect your relative risk ratio. But the way we calculate it is the prevalence of disease in the relatives of affected persons divided by the prevalence of disease in the general population. So higher values for the relative risk ratio indicate a larger proportion of risk in the family compared to the population. This value increases with the increasing genetic contribution, and it can also increase as the prevalence in the general population decreases, right? So it's a relative measure, and that's very important to bear in mind because without the knowledge of the population level risk, you cannot calculate this measure. So here are two seminal examples of calculator of relative risks. The first is cystic fibrosis. We now know that this is a disease that has strong genetic etiology. But here are how the data were considered. So the risk of the CF in siblings was 0.25 based on a myriad of data or studies. And then the calculated risk in just the general population of people, regardless of their family membership, was 0.0004. So the ratio of those two, again, this should be a lambda sign, is 635. So that's very large, suggesting a strong genetic etiology for cystic fibrosis. And then similarly, for Huntington disease, the risk in siblings is 0.5, a 50% risk, versus a very low risk in the general population. And that results in a relative risk ratio of 5,000. And this is just to illustrate the difference between someone who has a normal basal ganglia and someone who has Huntington disease and how their basal ganglia looks. And similarly, up here is the image for someone with cystic fibrosis, which is a disease of the lungs, and someone with a healthy lung.